pleasure to welcome all of you to this panel event organized by Africans in STEM and the Department of Pharmacology Graduate Committee. So this event is aimed at spotlighting Black female scientists in Cambridge and here in the UK to celebrate Black History Month. So I hope we have a wonderful hour ahead of us and we get to learn from each other as well. So my name is Charity and I'm a third-year PhD student in the Department of Pharmacology. I'm a member of the Balma Lab group and I'm also part of the Department of Pharmacology PhD and Postdoc Committee. I'm originally from Zimbabwe and I'm a member of Newnham College. So to begin, to kickstart our event, we're going to have the head of the Pharmacology Department, Laura Itzhaki, who will give us a few words and welcome everyone to this event. Thank you so much, Laura, for joining us, and thank you so much for your support to us this event. You can go ahead.
Great, thanks, Charity. So it gives me great pleasure to welcome everyone, albeit virtually, to the Department of Pharmacology, and we're very pleased to be hosting this event today, which is a spotlight on Black female scientists in Cambridge as part of Black History Month. So I'd just like to briefly say the event was organised by two very energetic individuals in the department. The first was Cynthia Okoye, who is a PhD student in the department and co-founder of Africans in STEM. And the second is Camilla Ascanelli, who's also a PhD student in the department and a member of the department's Graduate and Postdoc Committee. So just before I hand back to the panel, I'd like to do a shout out for the exhibition showcasing notable Black scientists from around the world. So this exhibition is, again, is part of Black History Month, has been rotating around departments. It was in pharmacology a few weeks ago. It's now in chemical engineering, but it's also available online, and that's till the end of the month. So without any further ado, I'll pass you back to Charity.
Sorry, I was mute. Thank you so much, Laura, for being here, and thank you so much for your words. And so now we'll start our event and we'll hear from our panelists. And so to begin, the first part of our event, we'll have each of the panelists give a 5-minute presentation about their research, and we'll take one question after their presentations. To all the people who are listening to this, could you please send in your questions in the chat, and then we'll pick one question after each presentation and answer that question for you. And then for the rest of the questions, please just hold on to them, and then we'll have a general Q&A at the end that we'll get to. Okay, so to begin, as I mentioned, we'll have 5-minute presentations from our panelists, and the first panelist who'll present their research to us today is Dr. Pisa Atakpa. Dr. Pisa Atakpa grew up in central Nigeria, and about 10 years ago, she moved to Liverpool to pursue an undergraduate degree in pharmacology. Having thoroughly enjoyed this time, she knew she had to pursue a PhD in the same discipline. So Dr. Atakpa completed a PhD in pharmacology in Cambridge, which was funded by the Cambridge European and International Trust. She now holds a research fellowship with Emmanuel College to study the dynamics of intracellular signaling. And furthermore, Dr. Atakpa is passionate about breaking barriers and inspiring the evolution of STEM subjects in Africa and among Black and ethnic minority groups. Thank you so much, Dr. Atakpa, for being here with us today. Please feel free to share your screen and present your research to the audience.
I'll just share screen. Thank you very much for the very kind introduction, and thank you to the organizers for inviting me and organizing such an amazing event in commemoration of Black History Month. Like Charity mentioned, I only have 5 minutes, so I better crack on. I am now a research fellow with Emmanuel College, and my research interest focuses on calcium signaling in the human body. We all appreciate calcium to be important for having healthy bones and teeth, but calcium, we now know, regulates a plethora of different processes, from memory to gene synthesis, development, migration, muscle contraction, and even coronavirus entry. Cells do this by having specialized pathways that are very tightly regulated for calcium release and sequestration. A very quick summary is that cells are usually protected by a plasma membrane that separates them from the outside world. And through different proteins, you can have signals from the extracellular space transduced into the intracellular domains of a cell. The endoplasmic reticulum is the most characterized calcium store in cells, and it's able to concentrate calcium into its lumen using SERCA, which utilizing the energy from ATP hydrolysis pumps calcium into the ER lumen. Now, upon stimulation of receptors such as the histamine receptor, you have the production of IP3, which acts on this receptor this is the most characterized calcium releasing protein that sits on the ER. It is called the IP3 receptor. Upon binding to its ligand, it releases calcium into the cytosol, and the effects of this cytosol includes the contraction and fertilization and the rest that I mentioned earlier. Now, this has been studied for many, many years, but my interest lied in these organelles called lysosomes. Now, lysosomes for a long time were thought to literally be the garbage can of cells where they digest unwanted materials. But now we know that they're actually more sophisticated than that. For example, virus entry into cells utilizes the lysosomal pathway. For example, some of the drugs used to target coronavirus actually targets lysosomes. We know that the calcium content of lysosomes are important for all of these functions, but we didn't know how this calcium entered into lysosomes in the first place. And this was the project I dealt with in my PhD. To cut a very long story short, what I did was using high resolution microscopy, I was able to look at the ER, that's the most characterized calcium store that I told you in the beginning, and lysosomes. So the video here shows the ER in blue and lysosomes in red. Now, so I'll play the video again. If you look at multiple examples, you can see that lysosomes sit at this ER junctions. They like to be closely affiliated with the ER. They traffic along the tubules for long distances, jumping from one part of the ER to another. And through many experiments that I'm not able to show you now, we were able to come up with this model. What we found was that lysosomes would sit at specialized junctions with the ER, which are usually populated by the IP3 receptors. What this allows is when calcium gets concentrated in the ER through SERCA, then these junctions provide an environment where calcium can then be released in high amounts to these junctions, allowing what could be a low affinity uptake system to then take up the calcium into lysosomes. This work was published a couple of years ago now, and I've gone on to look at, I'm still asking what the uptake protein is that actually allows this calcium to enter lysosomes. I'm also looking at what proteins regulate these junctions. Why are lysosomes able to stay at these junctions for prolonged amount of times? We now have some candidates we're going with, and I don't have time to go through all of that now. But that's what I do. And a very, very quick thank you to people that fund the work, and my supervisor, Professor Colin Taylor, that has been a great mentor throughout my PhD and continues to be my mentor now in my postdoc. And thank you very much for listening. I'll be happy to take any questions. I can stop sharing.
Audience members, please feel free to post your questions in the chat. And while we're waiting for questions to come up, I'll ask a question. Thank you so much for your presentation, Piers. That was very informative and exciting. I don't know if I missed this, but you mentioned that there are a lot of real-world implications with calcium signaling. So basically, how does your research or what you're doing translate into the real world, or like the bench, and how can we utilize that?
Thank you, Charity, for your question. So a very, I guess, recent example that comes to mind is understanding virus processing in lysosomes for coronavirus, for example. So understanding how calcium goes into lysosomes in the first place would allow us to know how we can modulate lysosomal function in disease, for example. Now, chloroquine, which is very contentious, has been used in treating some coronavirus patients. Now, it works by stopping lysosomal function. We know that it would stop calcium accumulation into lysosomes, for example. So my work is very basic science that we hope by understanding that it can then inform, you know, more medical approaches down the line. So, yeah.
Thank you so much. We have a few questions that came in for you in the chat, but we'll ask those towards the end.
Okay.
Thank you so much. Yeah. Okay. Thank you so much, Dr. Atakpa, for her wonderful presentation, very informative and clear talk. And next, we're going to have our second panelist. And our next panelist is Dr. Jerelle Joseph. Dr. Jerelle Joseph is from Dominica. She obtained a Bachelor of Science in Chemistry and Mathematics and an MPhil in Chemistry at the University of the West Indies in Barbados. She then completed her PhD in Chemistry at the University of Cambridge, and currently Dr. Joseph is a Junior Research Fellow in Physical and Chemical Sciences at King's College, Cambridge. Her research encompasses developing multi-scale modeling approaches to study how the cell organizes its contents via liquid to liquid phase separation and to examine the role of this process in biological function and malfunction. Alongside her research, she's the founder of Carri Scholar, an organization that connects Caribbean students, professionals, and academics in their field of study. Thank you so much, Dr. Jerelle Joseph, for joining us today, and we look forward to hearing your talk.
Thank you very much. Can everyone hear me? Thank you very much, Charity, and to the organizers of this event for having me. So when I speak to people about my research, I like to get them thinking about vinaigrette salad dressing. Yes, you heard right, vinaigrette salad dressing. So if you ever had vinaigrette salad dressing, you may have noticed oil droplets suspended in the vinegar. Oil and vinegar are both liquids. However, when mixed together, they tend to separate into two liquid phases. And this process is known as liquid-liquid phase separation. Interestingly, it was recently discovered that liquid-liquid phase separation occurs inside cells. So proteins and other molecules that are generally dissolved in the cytoplasm and nucleoplasm, they can actually interact with each other and condense, forming these oil-like droplets in cells. These droplets are more formally referred to as biomolecular condensates, and they are believed to play critical roles inside cells. For example, the formation of biomolecular condensates has been proposed as a key mechanism used by cells to organize their genetic material. So certain proteins have been shown to actually bind to DNA, compacting and sequestering it within distinct liquid-like compartments. And in doing so, this condensate, if you think about it, by having DNA inside of these compartments, this condensate actually regulates the way in which our genetic code is accessed and determine which parts of the genome are expressed. Importantly, biomolecular condensates may mature and transform from their normal liquid-like states into solid-like aggregates that hinder proper cellular function. For instance, several protein aggregates that have been implicated in neurodegenerative diseases have also been shown to, these proteins that form these aggregates have been shown to form biomolecular condensates as well. So it is therefore a matter of both broad and urgent scientific interest that we understand what are the molecular mechanisms that govern the formation and dissolution of biomolecular condensates. So some key questions that we ask ourselves are, what are the defining characteristics of molecules that undergo liquid-liquid phase separation inside cells and make up these condensates? What dictates which parts of the genome are included or excluded from biomolecular condensates? And how do molecules such as DNA behave once inside condensates? And importantly, what causes useful liquid-like cellular compartments to mature into pathological aggregates? So investigating liquid-liquid phase separation in cells has proved, however, to be quite a challenge, because on one hand, experimental techniques can mainly provide low-resolution data for the larger systems involved, and on the other hand, standard computer simulations can provide us with intricate details, but mainly for the smallest molecules involved. So we therefore require biophysical approaches that can provide us with the missing close-up views. My research precisely targets these challenges by developing multi-scale computational approaches to probe the formation of biomolecular condensates via liquid-liquid phase separation. So these multi-scale approaches that I speak of, this involves developing minimalist computer models that can allow us to simulate tens of thousands of molecules at moderate computational costs. And in such simulations, we can conveniently vary numerous properties of intracellular mixtures, such as the concentration of molecules and the binding strengths of molecular components.
Hi, Jerelle, sorry to disturb you. Do you have a screen to share?
No, I don't.
Okay. So sorry, we were wondering if there's a screen that we're supposed to be looking at. Sorry.
No.
Okay.
Hence, we can obtain a set of general rules for systems that undergo liquid-liquid phase separation. And I then use these rules that I get from the minimalist models to inform the design of simulations that are conducted at higher resolution, so with more details in my simulations. And using this sort of simulations, the chemical makeup of specific molecules can be represented, and we can also represent intracellular salt effects and other environmental conditions that may be present inside of the cell that are important for liquid-liquid phase separation. And then combining these simulations, we can determine the precise interactions that dictate how molecules inside cells partition into different liquid phases. So my work is coupled with experimental collaborators, and our research not only focuses on extending our current understanding on this new phase of intracellular organization, but also we're trying to be able to develop models that can inform the design of reactive volumes, that can propose routes for bypassing aggregation-related disorders, and ultimately to help bridge the long-standing knowledge gap between what is the genetic code and how precisely it behaves. Thank you.
Thank you so much, Jerelle, for sharing your science with us. We really appreciate it. Thank you so much for being here. Audience members, if you have any questions, please feel free to send them in the Q&A or in the chats, and they can be answered right now throughout the panel, or we can get to them at the end. So while the questions are coming in, thank you so much for the very interesting talk. What was the most challenging, possibly technique or experiments that you had to do during a PhD? I know you got your PhD recently, not very recent, but how did you overcome any challenges in your experiments, and how did you get through it till the end?
Yes, so I am primarily a computational theoretical chemist. So I do not do any wet lab experiments. I discovered very early in my undergrad that I did not like the lab, and I was very clumsy. So in terms of challenges during my PhD, I think it's not to do with one particular thing, but more so in terms of the PhD, it had more to do with endurance and being able to pace myself properly while doing my work, and recognizing that the PhD was not a sprint, was actually a marathon, and that I should keep at it. So in doing computational work, we always say that if you, and I know experimentalists would have their own timelines, but if you assume that something is going to take 6 weeks, it's probably going to take 6 months. So I think sort of being realistic as to knowing that different things can go wrong along the way and being able to adapt and be flexible for when things go wrong. Yeah.
Okay, thank you so much. So we have a question for you in the chat. Whenever you get a chance, feel free to take a look at that and then answer. And thank you so much for your talk. Thank you so much for being here. We appreciate it. Okay. We're going to move on to the third speaker of the day, and our next speaker is Stephanie Adeyemo. Stephanie is a fourth-year PhD student in electrical engineering at Hughes Hall, University of Cambridge. The key focus of her research is utilizing tetrahed spectroscopy, a contactless and non-destructive optical characterization technique to examine the optical and electronic properties of semiconductor nanomaterials, particularly of tin-based metal halides, perovskites, and nanowires. This gives insights into the fundamental physical mechanisms in these materials that guide device development for applications, including photovoltaics and photodetectors. She holds a Bachelor of Engineering in Electronic and Electrical Engineering from Swansea University, and her research is supported by EPSRC DTP studentship. Thank you so much, Stephanie, for being here. Please feel free to share your screen and present your research to us.
Right, thank you very much, Charity. So I would start sharing. Okay, so thank you very much. Thank you to the organizers as well for allowing me to be part of today's talk. So today I'm talking on time-resolved terahertz spectroscopy, and I'll go on. So first of all, the problems with, so first of all, nanomaterials. I work with nanomaterials, and the problem with these nanomaterials is that it's a good thing that their dimensions are really low and small, and the dimensions are about 1 billionth of a meter, and compared to a human hair is like 100,000 nanometers. So these materials are really tiny, and they pose, and when they go down to these dimensions, their properties change, and they are better for nanoscale devices as well. So that's why we go for, one of the reasons we go for nanomaterials. And the problem now is that there are very few measurement techniques. There are measurement techniques available to measure materials, but when it comes to nanomaterials, contact is a problem. And this, like for example, in this picture here, it requires additional techniques, fabrication techniques to make this contact, and sometimes this contact can pose problems to the actual results you're actually getting. So so today I'm talking about my own measurement technique, which is called time-resolved terahertz spectroscopy, and it overcomes these challenges of contact problems and destructive problems. So in this technique, we use laser light, short pulses of laser light we hit short pulses of laser light on the material we're measuring, and the electrons in these materials gain energy, and they become conductive, and then we can measure the properties of these materials, like how fast they move, how long they take to relax back to their previous state. So all these properties are interesting and important for if you are developing devices, if you are looking for best techniques for your best materials for your device. So all these properties are important, and no matter how fast or how slow the electrons are, they can be used for different kind of devices, applications. So with terahertz, why do we use terahertz? Terahertz energy is so low, and the energy is, so when we photoexcite, and then so in this technique, we photoexcite the material and then we probe with the terahertz. So terahertz has a low energy, and when we probe with the terahertz, it doesn't really photoexcite. It doesn't photoexcite the sample as high as before. And then also terahertz can also stimulate the motion of other kind of particles within the material, and because the terahertz time terahertz frequency scale is around the scale of the scattering rate of these electrons. So it's easy for us to identify and to probe of the electrons in these materials, properties of electrons in these materials. So like in this picture here, this is to excite the sample. In this picture, it's a nanowires on the substrate, and then at a later time, we then probe with a terahertz probe. And this is a picture of me in the lab, just to understand that just to understand what the setup is like, and we use optics to direct our laser light towards the material. So I'm just going to quickly explain this pump probe technique. So for example, I'm going to use a normal daily life technique. For example, if we know that this golfer is so talented, and then he can swing his ball so hard and consistently at 160 kilometers per hour, and then we have a photographer that wants to understand how the swing affects the ball projection. So one thing the photographer can do is that he can run through. Once the golfer swings the ball, he can run through and start taking pictures of the ball, or he can, because we know that this golfer consistently swings his ball at 160 kilometers per hour. What he can do is let the golfer swing his ball so many times, and then the photographer can move and take picture at different time delays. So this is what this pump probe technique is like. So in this sense, the golfer is the pump, is the one shooting the balls, and the photographer is the probe, is the one taking the probing the material or the ball at different time delay. So this is a simple illustration of what this technique is like. So we understand, so we are able to probe the different fundamental mechanisms that are going on in this material, like how fast we can understand the process the electron takes to go back to its previous state, to recombine back to its previous state. We can understand other kind of particles that are available in this, that are being excited in these materials. All those kind of things are important. So this is just a simple illustration of what the system is like. And then just going there, finally, I'll just briefly talk about terahertz spectroscopy of allied perovskites. So I mainly work with allied perovskites, and it has an ABX tree crystal structure, and perovskites are one of the most bubbly materials now when it comes to solar cells, because they can easily be tuned. So their band gap can easily be tuned across the solar spectrum, and it absorbs broadly across the solar spectrum. Another thing is that it's a solution process fabrication technique, which is very simple, low cost, and simple to easily fabricate even in your home if you have the basic equipment. And so with my terahertz spectroscopy, I look at these materials. We tune different things we tune different properties of these materials, like passivating it, looking at how it would change over time in, let's say, degradation, or how it would change over time when it's exposed to light, because obviously the end process of this perovskite will be under solar cell. So we understand all these kind of processes that are occurring within the material, and then we use it as a feedback process into fabricating solar cells. So my work is mainly into understanding these fundamental mechanisms. So thank you very much. Thank you for everyone listening, and thanks to also my sponsors, EPSRC, and thank you to African in STEM. Thank you very much.
Thank you so much, Stephanie, for your presentation. We really appreciate you being here. So please feel free to add questions to the chat or to the Q&A, and they'll be answered as we go. So just for the sake of time, we'll have just move our questions to the end, or just keep adding questions throughout the presentations, and the presenters will answer your questions as we go. And so we'll move on to our next speaker. And just a friendly reminder that let's try and keep the presentations 5 minutes so we can have everyone present in a timely manner. Thank you. So our next presenter is Tapoka Mkandawire. Tapoka Mkandawire is a student at the Wellcome-Sanger Trust Institute. And she's originally from Malawi, a small country in Southern Africa, and her second home is the north of England. Tapoka grew up in Liverpool and used to live in Manchester, and she's passionate about science as well as effective communication of science and the positive impact this can have on society. Tapoka works with the human gut, which is a hive of activity with lots of different individuals working together. All these activities have different effects and can make us healthy or feel unwell. The individuals she's interested in the guts are the whipworm and bacteria. Whipworm affects millions of people in tropical countries and can make children very ill. The gut bacteria talk to the worms, and so understanding what messages are shared between the guts and the bacteria will be helpful for beating this disease. When she's not experimenting in the lab, she's experimenting with food, trying new recipes or restaurants. Tapoka also likes to get out in nature for a good long walk or stay indoors with a good book. Thank you so much, Tapoka, for being here. Please feel free to share screen and present your science to us.
Thank you so much for having me. Don't have any slides, but we'll just get into talking a bit about the gut microbiome and parasites. So I study whipworms, which are nematodes, so roundworms. These infect around 20 different species of animals, which includes humans. In humans, this disease is called one of the neglected tropical diseases. So it's a disease that has a large impact on lots of people's lives. Around 400 million people are infected, but often this disease kind of goes unnoticed and underfunded when it comes to scientific research. That's why it's called neglected. So I want to understand how this disease infects humans. And to do this, I study the human parasite, but I also study the parasite that infects mice and the parasite that infects pigs. So the whipworm life cycle starts with eggs that persist in the environment. So you can find them in contaminated food or contaminated water sources. The host will come along and eat or drink this contaminated object, and then the eggs go into the intestines. The eggs hatch in the intestinal environment, and then the worms swim out and burrow into the intestinal lining. The worms grow up in the gut environment, and then eventually the host passes eggs back into the environment, and this completes the life cycle. It's what we would call fecal-oral transmission. Now, in your gut, it's a very kind of active environment. There's lots of different things that are there. So you have parasites, you have different bacteria, you have viruses. Now, it's very important for this parasite to know that it is in the correct gut environment for it to hatch. And one of the things that we think provide the signals for this is the gut bacteria that you have inside your guts. Now, these worms are smart in doing this. They don't just hatch in any gut environment. If you have the mouse whipworm, it has to hatch in the mouse gut. If you have the human whipworm, it has to hatch in a human gut. So this is also interesting to understand these unique things that are unique to each species of the worm, but there's also got to be some things that are common across the worm species because they all like guts. So yeah, in my PhD, I've been working with mice samples, with pigs, and with humans, trying to kind of drill down into what bacteria are important and what makes these bacteria important or special. What molecules and proteins do they have that the whipworm is looking for to hatch? Now, we want to do this for two main reasons. The first reason, like I said, this disease affects hundreds of millions of people worldwide. While it is a disease that is linked to water, sanitation, and hygiene, this doesn't mean that we can't find ways of treating people to try and beat this disease while we also tackle the infrastructure and socioeconomic problems. So understanding hatching and the basic biology of whipworms will help us to do that. The other reason why we want to understand these worms is because they are kind of a true parasite. They get inside the body, and for the most part, in adults that have chronic infections, they can live for years with this infection. It doesn't affect them too badly. This is because the parasite is very clever at manipulating the immune system, and it kind of tells the body to ignore the parasite and pretend that there's nothing going on. Now, in countries where they've built up this infrastructure around water, sanitation, and hygiene, we've also seen an increase in autoimmune disorders. So things like irritable bowel syndrome, ulcerative colitis, celiac disease, all of these diseases where essentially we think the immune system is not challenged enough and is kind of starting to get bored. So understanding a parasite that has this beautiful control of the immune system and learning how we can harness that will also help us treat those diseases in the future and kind of help us understand better what components of the gut microbiome are important for keeping away in terms of staying healthy and avoiding disease, but also that we maybe need to maintain to avoid these other diseases of the immune system. So yeah, I hope that piques your interest into whipworms, parasites, and the microbiome. And thank you for listening. Thanks to the organizers for inviting me, and thanks to my funders, the Wellcome Trust. I will take any questions.
Thank you so much, Tapoka, for your presentation. I really appreciate you being here. Audience members, please feel free to post your questions in the chat or in the Q&A, and we can get to them as we go, or we can get to them at the end where there's a general Q&A. We're going to move on to our fifth speaker of the event, and our next speaker is Dr. Ebele Mogo. Dr. Ebele Mogo is a researcher at the GDAR network of the MRC Epidemiology Unit of the University of Cambridge, when she leads investigations into how cities in Africa and the Caribbean are changing, how this shapes health behaviors, and leverage points that exist for pivoting this context to prevent non-communicable diseases. She also leads Engage Africa Foundation, a network on a mission to promote putting health at the center of development in light of the rise of non-communicable diseases. Dr. Mogo is a next Einstein Fellow of the African Institute of Mathematical Sciences. Thank you so much for being here, Dr. Mogo, and I see you've already shared your screen, so please feel free to share your science with us.
Absolutely. Thank you, everyone. I really enjoyed hearing a bit about everybody's research, so it's really fantastic. My name is Ebele Mogo, and I am a research associate at the Global Diet and Activity Research Network. I almost feel like Charity has said basically what I do. So I'm just going to share very quickly in 5 minutes a bit more. So really, there's a lot of things that are happening in cities and emerging economies around the world that have a lot of relevance for health outcomes. So you think about rapid urbanization, you think about climate change, you think about environmental degradation, rapid population growth, especially in African cities where you're having a very young population. It's the median age is about 19 years old. And so when we're thinking about health, you very quickly start to see that if you want to understand health, you have to understand all these dynamics and how they relate to how people behave in cities and then what becomes their eventual health outcomes. So a lot of the work I do essentially does that. It tries to do, it's a lot of transdisciplinary collaborations to understand how cities are changing, understanding how these different factors shape people's health behaviors, especially things like diet and physical activity, because they intersect with just so many diseases like diabetes, cardiovascular disease, and then trying to use that evidence to inform policy, inform the design of intervention, so that essentially urbanization doesn't come at the expense of good health and well-being. So I'm going to talk a bit about some of the work I'm currently doing right now. So I work as part of a collaborative, it's called the GDAR network, the Global Diet and Activity Research Network. And we have a lot of institutions from South Africa, Cameroon, Kenya, Jamaica, all looking at and comparing how the diet and activity contexts are in these different countries. So there are 3 key parts of what we do. One part is spatial epidemiology, just trying to characterize these cities. For example, in a city like Kisumu, what happens when you have a new grocery store? How is it going to shape people's dietary behaviors? How is it going to shape their attitudes to healthy eating? What are travel patterns like? How is it shaping exposure to things like pollution or injury, obesity? That's one part of it, just trying to gather evidence on the ground. Another part of it is trying to synthesize evidence on what has worked or what could work, because if we understand maybe some intervention that's happened in, say, Rwanda, maybe that has something to teach us about what we could do to improve public health in Nigeria. And then the third part of that is using this information to work with communities and decision makers to design interventions and inform policy, and they're all sort of related. So I'm going to talk a bit about some of the ongoing projects I'm working on right now. This is just a few of them, but one of the projects I'm leading is a systematic review. So we're trying to look at what interventions have actually aimed to improve health outcomes that have had different sectors working together. Because like I said, for you to improve something like diet, you have to look at transport, you have to look at urban planning, you have to look at the environment. So one of the reviews I'm leading is just trying to gather evidence of what has been done before and what can this tell us about designing interventions with different sectors collaborating. Another one I'm working on is called the Alpha Study, and it's looking at physical activity in public spaces and how people are navigating public spaces. And how does that shape, for example, if you go on a run in a city like Lagos, and you're probably going to run to improve your health, but how is that, how does that relate to your exposure to something like pollution? How does that shape your exposure to injury? And then this involves triangulating a whole lot of information. So we have citizen scientists, that are people that are actually running in public spaces, for example, gathering evidence from them on how on their cities, gathering evidence from the literature around sort of public spaces and the connection to health promotion, and then doing air quality monitoring in different specific spots in the different cities, and then integrating all this information together so that we can inform how we should be thinking about investing in public spaces if we want to improve public health. Another really interesting one we're working on, which is a new project, is looking at, is a social media analysis. So we're looking at basically what people are tweeting and trying to understand how are people sentimental around physical activity, health, and well-being. What do they look like before COVID and after COVID? Because a lot of African countries took a lot of measures during lockdown, for example, and they were all different measures. So like somewhere like Lagos had something different from somewhere like Nairobi and all of that. So we want to see what does the public think about this? What were their perceptions of their health behaviors like physical activity, as well as their perceptions of some of the control measures that were taken? Because we know that a lot of what is going on now has implications for how we deal with health crisis, not only just with COVID, but emerging pandemics that might come up at different points in time. So those are some of the things I'm doing. So the work I do is very transdisciplinary. You're always working with experts from different fields, and you're always working in that space between sort of research, linking research to action, linking research to policy, linking research to interventions. So that's it in a nutshell. I look forward to answering any questions that anyone else has. I just want to say a quick thank you to all the organizers, from Camilla to Cynthia, to Africans in STEM and the Department of Pharmacology, to my colleagues at the JIDA group and everyone present. And today is African Human Rights Day. So I just wanted to also just share my solidarity with a lot of things happening. I'm Nigerian, and there's been a lot going on with the NSAS movement. So I just wanted to share also my solidarity with a lot of the movements that are happening as well. Thank you very much, and I look forward to the questions.
Thank you so much for your presentation, Dr. Ebele, Mogo. We really appreciate you being here and enjoyed learning more about your research. To all the participants, please feel free to send questions in the chats or in the Q&A section. And if you can, Dr. Ebele, could you please take a look at the questions as we go through, and we'll get to a Q&A at the end where you can also answer those questions live. Okay, so now we'll move on to the final speaker of this section, and our final speaker is Dr. Odiri Eneje. Dr. Odiri Eneje studied medicine at Imperial College London. She completed a Bachelor of Science in Medical Management at Imperial College Business School, and she then spent 4 years training as a junior doctor in the North West Thames Deanery and rotated around a few hospitals in London, including St. Mary's Hospital, Charing Cross Hospital, Hammersmith Hospital, and the Royal Brompton and Harefield Hospital. After her core medical training, she completed the Diploma of Tropical Medicine and Public Health at the Liverpool School of Tropical Medicine. She then traveled to work in South Africa, caring for patients with complex respiratory infections, including TB and HIV co-infection. On her return, she was successful in gaining higher specialty training in respiratory medicine in the East of England Deanery. She studied and was awarded a master's degree in medical education from the University from the University College London before joining the Flutter Research Group at the MRC Lab of Molecular Biome. Her interests are in complex respiratory infections and medical education, and the current PhD research focuses on exploring drivers of infection and inflammation within the context of cystic fibrosis. Thank you so much for being here today, Dr. Odiri and Eneje. Please feel free to share your screen if you have one, and we look forward to hearing about your science.
Great, thank you for that introduction, and thank you to the organisers for inviting me to speak. So my name's Odiri Eneje. I'm a medical doctor and also a PhD student at the Lab of Molecular Biology. So I'm going to talk about my research project and then also just spend one slide talking about my journey and how I got here. So my overall broad scheme is looking at tackling inflammation within the context of a disease called cystic fibrosis. So these 4 panels represent an overview of my project. So cystic fibrosis is a genetic disorder and it affects many organs in the body. I focused in on the lungs because that's the organ that causes the most amount of disease and comorbidity to our patients. And they suffer from shortness of breath, cough, sputum production, and recurrent infections. If we move to panel 2 and zoom down to a cellular level, the gene that codes for cystic fibrosis codes for a protein called the cystic fibrosis transmembrane receptor. So this is an ion channel that moves chloride, represented as Cl-, out of the cells. In cystic fibrosis, this doesn't work. And so chloride stays inside the cells. With chloride staying inside the cells, sodium, represented as Na+, stays inside the cells. And with sodium staying inside the cells, also water, represented as H2O, also stays inside the cells. This results to drying out of the surface outside the cells. So if we go to panel 3, we can see some normal lung airways. And if you can see that they're nice and smooth and clear. On the right-hand side, we can see some CF airways.
Excuse me, sorry, Dr. Odiri. We're not seeing your slides move on. We're just seeing the first slide. Is that
okay? Yeah, no, that's fine because they're in 4 panels. So panel 3 at the bottom.
Oh, okay, thank you.
Yeah. So in the CF panel, in the CF airways, you can see lots of mucus. So we breathe bacteria in and out of our lungs all the time. And if you go to panel 4, you can see that the body produces the innate immune system causes a response, which also then the adaptive immune system comes in, and it causes cells to move to the area of infection. And that's what we call inflammation. In the acute phase, that's good, it helps clear bacteria. However, if it persists, you get damage of the structure of the walls, and then that causes you to be more susceptible to infection, which causes more inflammatory mediators, more cytokine production and more damage. Eventually, it results in tissue damage and remodeling. So that's an overview of the problem. Next, I'm just going to show you a slide of some lung airways. So these are little cells that have.
Seem to have again. We're still not seeing anything, and we didn't see the panels as well at the bottom. Is that?
Okay. Let me just stop sharing for a minute.
Thank you. I'm really sorry for interrupting.
That's okay. I'm not sure why that's happened.
Oh yes, now we can see something.
Okay. So should I try this now? Can you see it now?
Yeah.
And can you see this?
Yes.
Okay, and can you see them beating?
Yeah.
You see the video?
No, it's just the one slide with the 4 panels right now.
I'm not quite sure why it's doing that, so I'll present it in this view then.
Okay.
So in this view, you can see there are cells of the lung airways, and they beat. And what they do is that they beat and they move mucus, as you can see those globules moving down. Up out of the airways so that we can cough it up. So we treated the cells with a phosphodiesterase inhibitor to see if it makes them beat better and also beat in coordination. And we did that in collaboration with the physics department. So then the next 2 are, we then gave these drugs to patients as part of a clinical trial. So the first slide shows actually, despite the fact, so this is a new drug that's nebulized, and that's the novel form. and we see that there's an increase in the drug concentrations, which is good. We also then measured factors that would impact the patient, i.e. how well do my lungs work after this. So we looked at spirometry, and we found that actually there were significant improvements in lung function after just one nebulized dose. So this is early study, it looks really promising, and more evaluation is ongoing. Another aspect of my project is that I'm genetically profiling the innate immune cells. So I take blood from patients that have cystic fibrosis and blood from patients that don't have cystic fibrosis. I then in the lab isolate the innate immune cells. So I look at neutrophils and I look at monocytes, and then I extract the DNA and RNA and sequence that to compare the differences. And I'm hoping that that will give us targets that we can look at targeting inflammation that way as well. So finally, just a quick slide about my journey. So studied medicine, then spent 4 years training as a medical doctor. At that point, I discovered my interest in lung infections. So did the diploma of tropical medicine and public health, and then spent some time working in South Africa before coming back to do a master's. I then joined the disease of chronic infections at Papworth, and then decided to undertake a PhD in that subject. So my tips are find something that you are passionate about, and I found that along my journey. Have a good mentor, and I had one. I could, at every stage of my career, I could tell I had a mentor, someone who was doing something that I was interested in, and also quite inspirational. And finally, self-belief. So thank you for listening. Lots of people that I work with. Thank you.
Thank you so much, Dr. Odiri and Eneje, for sharing your research with us. Thank you so much to all our panelists, and thank you so much to the audience members for being here and for engaging with our panelists. Please feel free to keep sharing questions, asking questions in the chat or in the Q&A, and then they'll be answered as we go, or we'll have a short section at the end where you can answer questions and they can be answered. And now we're going to move on to the next section of this event, and we're going to have a panel discussion with our panelists, and they'll share their general experiences of being here in Cambridge and being Black women scientists in the UK. So I'll ask a few questions, and then based on your experiences and whether if you have something else to add on to the conversation, please feel free to chime in into the conversation. So I'll begin. So my first question for you is, if one or two of you could just generally describe, your experience with being a Black scientist in Cambridge and the UK. And for those of you who've had the opportunity to learn in your home countries, how does that experience differ from the one that you've experienced here in the UK? So if anyone wants to start, I know, Jerelle, you did go to school in West Indies. So if you want to start.
Okay, thank you, Charity, for the question. So, well, I did my undergrad, as you said earlier, and my master's at the University of the West Indies in Barbados. And I'm not from Barbados, I'm from Dominica, but yes, same Caribbean. How was going to UE different from Cambridge? I think one of the main things is that UE is basically, well, especially University of West Indies, KVL, is like a Caribbean melting pot. So you find a lot of Caribbean people there, and it was diverse in terms of having different people from the Caribbean everywhere. And so the university is predominantly made up of Black students. So the language sort of that I would have been introduced to when I came to the UK would be like minority and so on. You would not use this sort of like categorizations at UWI because most people, not all, but most people are Black descent, of African descent. And so, yes, so it was coming, I believe when I speak about these things, I always have to put it in context in terms of I'm speaking as, again, somebody who is from the Caribbean, who grew up, spent my entire life in the Caribbean, and my sort of first sort of exposure to being in a non-predominantly Black society was when I came to Cambridge to do my PhD. So I never felt like I was one of the few when I was at UE, because everybody around me were basically like me. And then when I came over here to do my PhD, you kind of feel like, oh my God, I'm the only Black person in this meeting. I'm only the Black person here. And that stands out a lot. And I think it plays a lot in your psyche. And I did not realize that I had so much self-doubt until I came to Cambridge. And imposter syndrome, you hear everybody speak about it and stuff like that. And it took me a while to really find my place and find my community. And whereas when I went to UA, it's just like you started and everybody were friends like the first day. And I think it had to do a lot with me not putting myself out when I came to Cambridge. I was sort of to myself for my first year, because I was like, what did I get myself into? Like, why did I choose to come here? And do I really belong in this space? So it was a challenge mentally for me being in Cambridge. And yes, I guess many people would mention the word racism, and something that I would tell my friends is that I don't even know what I'm experiencing, if that is racism that people speak of. And I had a huge fear of like categorizing and saying that somebody was being racist towards me, because I felt like my radar for that was totally off, because I had never been placed in that sort of like space before. And I think all of these things sort of like, when I was in Barbados, I was 50 minutes away from home. So it was a 50-minute plane ride. When I was in the UK, I was so far away from home. So from like missing home, from being the only Black person in many spaces that you go, from feeling the pressure of being the first this, first that, and you feel like you really have to succeed, like you can't really like fail. All of these things sort of fit into imposter syndrome that you feel when you come here as, well, I'm speaking for myself as a Black person. And so I think it was very challenging, like mentally for me here to get my footing. But once I started to put myself out there and make friends and connect with people, then the experience was so much more rewarding. Yeah.
Thank you so much for your response, Jerelle. Dr. Peace, if you don't mind speaking to this, could you please just share what you think are some very apparent obstacles that Black scientists in Cambridge or in the UK generally face, and how those can be resolved?
Thank you, Charity, and thank you, Jerelle. That was a very good summary of the experiences of many Black people like myself, like ourselves, that I've heard in Cambridge. And I mean, I think that's, you asked me to comment on challenges. I think that is a huge challenge that many people have to initially go through, like in their first month or year, or however much it takes people to then, you know, settle into the work and, you know, shine. I definitely, for myself and many people I've spoken to, the minority syndrome, it allows the imposter syndrome to thrive, because if there are not many people like you in the room, you start to question yourself a little bit. How did I get here? I mean, for myself, somebody that was quite friendly with in my first year was joking and said, surely you only got into Cambridge because of a quota. And I internalized that for a while, and I was like, okay, maybe that's what I gotten. But then I had to remind myself, I graduated top of my undergraduate class. Surely I got here because I deserve to be here. So I think the lack of diversity, it's quite deep in the challenges that Black scientists have to deal with, because as a Black person, we come from like a different culture. So the way you express yourself is different and could be different, and sometimes may be interpreted for lack of intelligence or not being very confident. But that's just because people haven't, or some people haven't experienced diversity. They don't know how to communicate with you. So I think just that lack of diversity, just that minority syndrome is a huge challenge that Black scientists deal with. And there's sometimes lack of opportunities as well. I like to describe it as if everybody, interviewing you, or the panel that's choosing a candidate, if they don't look like you, so there's an unconscious bias that as a person you may not realize you have towards the kind of people you employ or you invite for talks. So I just think you almost have to do a little bit more to show your worth, just to break the glass ceilings. So I just think until we have more diversity, a lot of Black scientists will struggle almost to climb up that ladder. But I am so thrilled to hear from so many amazing speakers today that are shining and doing well regardless of the challenges. So I am really hopeful. I should end with that.
Thank you so much, Dr. Atakpa, for your response. And now I'll direct this question to Dr. Odiri Eneje and Dr. Ebele Mogo. I know both of you presented your work and it sort of fits back into the Caribbean and Africa. And so could you please tell us a little bit more about how your work is sort of, directly helping, or how is it related to hopefully possibly developing the African continent or the Caribbean, or improving Black lives everywhere? We could start with Dr. Ebele Mogo.
Okay. Thank you, Jerelle and Peace. I was smiling when you were talking. I was like, maybe I should have talked to you when I came to Cambridge. But talking about, I definitely, so my journey here has been sort of, I definitely have that sense of, I guess I would call it a passion. Like I really am interested in ensuring that the work I do has some sort of contribution, and it's sort of a great opportunity. It's a privilege to do that kind of work that I think has a potential to shape urbanization. A lot of the things that I'm studying are quite complex, and so you're looking at systems, you're looking at the built environment, you're looking at access to healthy food, you're looking at policies, you're looking at communities. So there's definitely that combination of obviously a strong desire to make a contribution, also a humility, because you know that a lot of these things take time. I think a lot of the work I do uses a systems lens. So trying to make sure that even from the beginning, when we're even thinking about how we're designing projects, we're trying as much as possible to do like a mapping of who is in this context, who is in Lagos, who is in Yaoundé, who is in Johannesburg, for example, so that understanding who are the key actors in all these spaces and trying to bring them into the creation, say, of the protocol. And then so that by the time that we're coming up with the research findings and all of that, we also have people that already know that this is going on. So we do a lot of work around stakeholder mapping, understanding who's playing in these spaces, who would this information be important for, what information would be useful to them, and how, so that the research can stand a chance of making a contribution in shaping interventions. What other work is going on the continent and in these different cities that maybe our work needs to piggyback on. So I would say that those are some ways that I hope that the work I'm doing can make a contribution. Charity had briefly mentioned some of the work that I'm also doing around more, not necessarily Cambridge related, but still in the same vein around sort of bringing together a group, a network of young people that are also passionate about ensuring that development is linked to health and well-being. So I find a lot of fulfillment from that as well. We do a lot of work around sort of trying to do more public engagement around what we're seeing in research, around development issues, and running competitions to engage young people around the connection between health and sort of the broader social determinants of health. So those are some of the things that I'm doing that I hope can contribute in a positive way to development.
Thank you so much for sharing that. We really appreciate it. Dr. Odiri and Eneje, do you want to go ahead and answer the question?
So thank you for the question. So I was born in Nigeria, and we have a lot of extended family that are still based in Nigeria. I spend a lot of time traveling between places. So I'm a medical doctor with lots of experience in respiratory health. So one thing that really was one of the reasons why I went into respiratory medicine is that I noticed that at home in Nigeria, we cook by over open stoves sometimes, and that causes a lot of respiratory problems in patients and also in young children. And so my time in the School of Tropical Medicine in Liverpool, they were doing quite interesting studies in Malawi, because they've got a base there where they were looking at a simple intervention where they did something where they gave a cook stove to see if that would improve outcomes. It actually didn't, but it got me thinking of actually how could I use my skills to impact respiratory health, because that's where my expertise lie. So my project's quite interesting because I've learned a lot of techniques. So it's been a combination of wet lab and also some bioinformatics. And I think those skills are easily transferable. There are people who are taking patients who have respiratory illnesses and looking at their genome. It's done quite commonly in the UK, and there are good projects that are going forward. And now actually, how do we do that more commonly in Africa? How do we expose more patients? How do we get patients involved in clinical trials, get them new medications that could be trialed in a safe and controlled way? So I'm hoping that by the time I finish my PhD, I'll be equipped with the experimental skills and also the bioinformatics skills. And then also, because I've done lots of work in regards to setting up clinical trials, looking at the ethics of clinical trials and things like that, I'm hoping that also that will feed back into how do we organise good, well-designed clinical trials that address a question in regards to respiratory health. So I'm hoping that that's what my research will go on to help with.
Thank you so much for your response and for your contribution. We really appreciate it. And then I'll have one more question towards Tapoka and Stephanie. So I know you're both doing PhDs, and the PhD can be very trying. So how do you overcome failures and challenges during a PhD, and what other things do you do outside of your research for socializing or fun? Tapoka, do you want to start?
Yeah, sure. So I think it's important just to admit that there are definitely going to be difficult days, and trying to get better at asking for help and trying to get better at just managing your workload and realizing you don't have to do everything all of the time, which kind of feeds back into that idea of feeling like you can't ever be a failure or anything like that. But it's just the nature of science. Sometimes things don't work and it's not really about you. So just, trying to keep that at the forefront of my mind. In terms of things outside of research, recently I had the pleasure of helping organize the Black in Microbiology Week. And that was an incredible event, which just kind of does the same thing, where it gets you in a room with loads of people who look like yourself. And this is people across the world. We had organizers from Australia to California. And it was just an incredible, affirming experience of all of these people working in a similar space to what I do. And just that is very encouraging. So that was the latest in my string of scientific outreach and public engagement.
Thank you. Stephanie, go ahead.
So thank you. So I think in my PhD, at the beginning of my PhD, just like Jerelle said, and Piers, you feel like you are the only one in that. Especially for me, because I'm doing electrical engineering, and even in electrical engineering, women are already minorities. So for me, at that moment, when women are minorities, and then Black women again are under section of minorities. So for me, when I first came in, there weren't many people to talk to when it comes to relaying my struggles, and sometimes you'd feel that you are the only one struggling, but whereas that's what a PhD is like. You would struggle. There are more failures and successes. There are more frustrations and successes. But when you get that success, it's going to be like there were never failures. So just keep on pushing, keep on, even if you are the only one, try to talk to people, try to look for mentors, try to ask questions, try to ask people especially people that are, let's say, a year ahead or 2 years ahead, ask them how they struggle with their first year. Just keep on asking questions, ask people about their experiences. Don't just talk about your research only, ask about your experiences as well. I think this platform is good, and this panel is good for people to actually realize that, yes, we face this kind of difficulties, these things are real. And for you not to feel that you're the only one experiencing that such kind of thing, you should also speak out, talk to people, get help, learn from other people. And then for me, in my apart from research, I do a lot of outreach work in Nigeria. My sister and I, we organize engineering outreach to schools in Nigeria. We go to schools, we talk to them about what engineering is like, and we also focus on girls, because one of our problems, as we see, women are less when it comes to science and engineering. So the main focus is on girls, because girls they will see that there are more men, just like I am in electrical engineering, there are more men in that field. But if you actually go there and they see women that are actually doing those kind of things, they would actually be more encouraged. And that was actually what helped me when I was younger in secondary school in Nigeria. I also attended this kind of event, and I saw women that were doing civil engineering as well, not just men, women, and all these kind of things encouraged me. So in my free time, I also try to do the same thing to other young girls, because I know how much it really helped me when I was younger as well.
Thank you so much. Thank you so much for your contribution and for sharing that with us. I'll ask one more question and then we'll wrap up the event. And so over the last few months, we've seen the Black Lives Matter movement gain traction, and we've seen protesters going to the streets in the US and all over the world. And so could I ask you ladies to just maybe make a comment about what the Black Lives Matter movement means to you? So if Jerelle, you could take that question, and then could I also have one of my Nigerian friends talk about the End SARS movement, because over the last 2 weeks, we've seen Nigerians take to the streets to protest police brutality. And last night, we saw that the police opened fire against unarmed civilians. So if someone could just speak on that and how that impacts you as, Black women here away from home when all this is happening. So if Jerelle, do you want to start and speak on the Black Lives Matter movement, and then someone else can speak about the End SARS movement that's currently happening in Nigeria? Thank you.
Yes. I don't know if there's a way to speak about this movement in few words, but I know for all of us Black people, this year has been very trying, and it has been an emotional toll on all of us and extremely draining. But I think that what has happened this year, because the movement didn't start this year, but the way things transcended this year in particular, I think it has the potential to have a long-lasting impact, way more than we have seen in previous years and in previous movements. And I see it in terms of like the response in different organizations and different departments trying to hold themselves accountable and trying to develop structures and put things in place to do better where it comes to anti-racism and diversity training and so on. So I think it is a very important movement, and I'm really hopeful, I would say, for the future and to see continued anti-racism work and so on. And I think that those of us who were sort of directly impacted by it in terms of like the emotional toll, I would advise you to take some time, you know, for yourself, and self-care is important. And I think so for somebody like me who doesn't work directly in sort of social issues, like I'm a scientist, right? I work on science problems. You feel the sort of like pressure to sort of like, you're supposed to be an expert in this, but I just want to like advise other people who are like myself to let you know that it is okay if you're not an expert in racism. And I think that you don't have to feel pressured to be the voice for all Black people. I think it is important for organizations and universities and so on to recognize that. So for example, if you want to build a good house, you get a good contractor to do it. So if you want to do good anti-racism work, sort out the people who have been working in this space for many years and have them do the work and help you to build these structures where your employees and where students can survive. And those of us who do not directly work in this place can feel that we can continue being the people that we are, that we can continue to do our science, and we can continue to do the things that that we want to spend our time doing, and that we don't have to feel pressured into being, all of us being like activists. So that's just something that I want to add on to that.
Thank you so much for your response. Dr. Ebele, I know you mentioned a little bit about this movement during your talk. So do you want to chime in and talk a little bit about the End SARS movement that's happening in Nigeria?
It's be hard to know what to say, but I think the summary of it is that we had a lot of young people protesting, a lot of primarily millennials, Generation Z. I said that they're protesting against police brutality. So you have this thing where there's a lot of stereotyping going on, you know, like I actually have, my hair is colored if I were to take off this scarf, and imagine walking down the street and someone say, oh, you're a criminal because your hair is colored, because you have a scar, or because you're a coder and you work on a computer, so maybe you're a scammer, you know, things like that. And I think that it's to me, it's quite connected to the broader Black Lives Matter movement, because I think that oftentimes we're also replicating some of these colonial ways of governance. And then for me, as a public health scientist who is saying, okay, well, how do we improve health? And how does the built environment shape health? And how can we do this in a context where we have a primarily young population, and we should be investing in this population if we want to improve health? Because this is, a lot of the brutality is connected to injury, it's connected to mental health, it's connected to long-term health. So I'm not quite sure what to say, because this happened yesterday, and I just, I think it's quite, it's just sad. I was very sad. And I was thinking about this event today, and I was like, oh God, am I going to be able to make this event today? And then I thought, okay, I'm going to be talking to Black people, and I would love to, you know, it's just be, and I'm happy I came, because it actually made my day. So I don't really know what to say, but I think that it's food for reflection, because you have to think about how many scientists, how many innovative thinkers have had their lives cut down because of these kinds of systems before they even get to the point of coming to university and doing a PhD and doing a postdoc and then being here. So I guess that's all I have to say. I don't know if the other Nigerian has some, other Nigerians have something else to add, but I think that's all I have to say.
Thank you so much for that response. Would anyone else on the panel want to say a few words about this movement?
Just to say, it is hugely emotionally draining, like Ebele mentioned, and we should just look after ourselves and, you know, pray, or, you know, if we have to switch off social media for a while, it's okay. Do whatever you need to do to look after yourself. And we just hope that, like the Ebele and Jerelle mentioned, the unity that has come of this movement, I hope it means that some true change in some form comes out of it, and that should almost be a consolation, I guess. And, you know, we hope for the best.
Thank you so much for your response. And now to wrap up our event, if any of the panelists have any last words, or I could ask you to say something to young girls who may be watching this and encourage them to get into science. So I'll give you each 10 seconds to say your final words of encouragement to other young women who want to get into science and let them know that it's possible. So we'll start with Dr. Peace, 10 seconds.
Oh God. Science is for everybody. I mean, if I can do it, that's what I say, anybody can. There are multiple opportunities out there, and I just wish young Black girls, ethnic minority, girls and boys, everyone, non-binary people could just access it and take those opportunities. But science is for everybody, and it's super cool. That's my thing.
Thank you so much. That was on time. Thank you. Next, we'll have Tapoka.
Yeah, I would say stay curious and always follow your passions. And that's how you, find yourself following some really interesting things. And the best things are often the things you are not expected.
Thank you so much, Tapoka. Stephanie?
I would say keep on looking for information. Because sometimes you wouldn't find Black women in places, even if the person there that is going to mentor you is not in science. Look for someone that's going to push you, that's going to even if the person is not in science, the person can still use their idea from whatever background they are in to push you, because you have to be there as a Black woman. You have to maybe be the pioneer of different things for Black women, so that other girls will also be drawn to it as well.
Thank you so much, Stephanie. Jerelle?
I will say to always be true to yourself. Never put yourself in any box that people try to force you into, and seek out people who inspire you and ask them for help, however you can.
Thank you, Jerelle. Ebele?
I would say that what you actually have to offer really matters. A lot of challenges we face today need different ways of thinking. They need different school knowledge systems, and they need what you have to offer. So it's not just about fitting in, but knowing that actually what you're bringing is something that hasn't been offered elsewhere, and the perspectives that you have can shape science. So you have something unique to add. It's not just about you fitting in, but it's about you shaping the science as well.
Thank you so much. Odiri?
And so, I guess my advice to young girls in situation is that I was once a young girl looking up and thinking, I love science. I like to know, why does the heart beat? Why do we breathe? What happens to your blood? And I think it's really having that passion, and don't let anybody tell you that you can't do it. You know, I had a teacher tell me, what, you want to do medicine? And I did it, and I love it. And I think the key is really to find what motivates you, find good role models, and believe you can do it.
Thank you so much to all our panelists, and thank you so much to all of you for being here in attendance of this event. I'd like to thank the Department of Pharmacology and the Department of Pharmacology Graduate Committee for helping us organize this event and for all their support and resources to put this together. Thank you so much to Africans in STEM for also organizing this event. Thank you so much to Camila and Cynthia for spearheading this effort as well. And thank you to all who joined today. I hope you have a wonderful rest of your evening, and I hope this event, you know, I learned a lot from it, and I hope you did too. It was very informative and fun, and it's very exciting and inspiring to see Black women doing amazing science in Cambridge. So thank you so much to all of you for being here. Thank you for inspiring all of us, and I hope good luck with all your research and life. Thank you.
Thank you.
Thank you.
Thanks.
Thank you.
Thank you.
Thank you very much.
Yeah, Cynthia, I don't know, do we end this?